Good afternoon, Professor Hoffmann, Professor Chiao, ladies and gentlemen. It's my great pleasure to be here to welcome you to this special dialogue entitled Exploring Innate Immunity Through the Lens of Fruit Fly Models. This event is particularly meaningful as it coincides with the 10th anniversary of the Hoffmann Institute, a center dedicated to advancing our understanding of innate immunity and fostering of esteemed scholars and a thought-provoking discussion. Today, we have the privilege of hosting some of the most distinguished minds in this field. We warmly welcome Professor Jules Hoffmann, a Nobel Laureate and pioneer in studying immune responses using fruit fly models, and it is also our editor-in-chief of Each Life. Alongside Professor Jiao, Professor Limner, Professor Faradon, and we're also delighted to have three talented young scientists here. Professor Hua Cai, Professor Di Chen, and Professor Chuan Xianwei. So fruit flies may be small, but the discoveries they have enabled are monumental, revolutionizing our understanding of innate immunity across all life forms. This discussion promises to uncover key insights, highlight current challenges, and illuminate the path forward for future research. Okay, let's begin the exciting conversation.
Sorry. So we'll be starting from me. I will ask the first question to Professor Hoffmann. So could you please tell us, in your mind, what is the historical milestone in the innate immunity research?
So let me first start saying that infections were one of the major problems for humanity after, let me say, probably when in the Neolithic period, people started to live in villages and in towns and so on. Just one example, in the valley of the Jiang River, and about 150 years ago, it has been recorded that people, young children, by the age of 5, 50% of them had died, and it was essentially of infections. And people, it was similar in other countries, and people didn't know why you would just get a fever then and eventually die off in large proportions. And this has changed. So what they thought was it was essentially punishment from the gods, and from you had done a mistake, or your country had done a mistake, your ruler had done a mistake, and you were punished by God. And that was particularly important during the Middle Ages and even before that, a century before. So what changed was the vision of two persons, really, two central persons, one in France, Louis Pasteur, and the other in Germany, Robert Koch. And around 1860, 1870, they found that the origin of these infections was microbial. And well, that was really, I would say, that was watershed. But it was not believed by everyone. And in addition to that, neither Pasteur nor Koch were wondering what do the animals or the humans, what reaction do they build up to fight now that they were convinced that it was microbial origin, how do they react? How do they react against this aggression? And that, the first time really, when someone asked a question and found an answer, that was in 1882, and that happened in Italy by an immigrant from Russia, whose name was Ilya Metchnikov. And he was working on sea urchins and on small invertebrate animals on the Strait of Messina. He was in Naples, Strait of Messina, separating from Sicily. And then it was probably by serendipity that he found that he was injecting several substances, particularly grains of colorant substances, into the transparent, translucid larvae of, essentially at that time it was the sea urchins. And he saw that small bodies in the blood were jumping on this and were trying to encapsulate, to integrate them and then degrade them. And he was very excited when he saw that, and he had the idea, that was really the idea of a genius, he had the idea, maybe the same is going to happen to bacteria. And then it's really worth, if you have the occasion, to read his biography. And he explains that he didn't sleep for a full night, waiting, he had injected in the evening bacteria marked by colorants, injected into sea urchins, and the next morning when he got up, he went to see the sea urchins, and yes, he noticed that this small corpuscle had engulfed the colorant, and they had to, in the process of degrading that. And then, well, this is probably the solution. This is probably what happens. And then he was extending over weeks and weeks, and even months, he was extending this to vertebrate animals, to other forms, and injecting various types of bacteria, fungal spores, and so on. And then he had, his second marriage was to a lady of Russian origin, but very familiar with Greek, ancient Greek. And he said, how shall we call those cells? And what do? She asked him, and he said, well, they eat, they are cells which eat foreign substances. And then she said, well, eat in Greek is phagein, and cell is kytos, so let's speak about phagocytes. Wow, he said, that's fine. And that was a moment when, it's somewhere in his memoir, it is written on which morning that happened, but that was the origin of phagocytosis. And so he was so excited, and he went and he was a professor at that time in Odessa, and then he went to the Pasteur Institute, and he gave many lectures on that, and surprisingly, several people, or I should say many people, were reluctant to believe that this was a solution to the diseases, to counter microbes. But okay, he was convinced, and he said, this is a solution to fight infections. But then, not far from there, well, not far from there, I mean, walking distances certainly, in Berlin, in Germany, a man named Behring, he had realized he was working on toxins, which are familiar to Dominique. And he could show that he was working on horses. If you injected, he was working with diphtheria toxin and then tetanus toxin. When you injected those into the big animals and you withdrew the blood after several time intervals, you would find something which was active against those toxins, and they lost their pathogenicity. And he called these toxins antitoxins, which later was replaced by antibodies. And then he worked with a Japanese colleague there, Kitosato. And then came Paul Ehrlich, also a German, who was close to Behring. And this is still in the end of the 19th century, beginning of the 20th century. And then Ehrlich proposed really a very deep explanation, saying that, okay, there are blood cells, and those blood cells, because those blood cells are responsible at a given moment for producing these antitoxins, antibodies. But he said, this is also, they are present in the plasma. Now the plasma has no blood cells. And the phagocytes of Metchnikoff were blood cells. And so there started the first controversy. And so Ehrlich said, no, it's not the story of phagocytes, that's not serious. Let's forget about that. And then started a period of disputes. And unfortunately, this happened at a time when there was very bad relationships between Germany and between France. And so the French scientists all were in favor of the antitoxins. And of course, the Germans, and the French side, following the Pasteur school, were in favor of phagocytosis. So this was a difficult situation in immunity, and when you look again at the history, in immunity in general, at the history, you will see that this has remained a problem for nearly a century. And according to the fact that you were German or from a Pasteur school, you would say this is important, this is not important. And this was annoying even for the committee of the Nobel Prize. They realized that these were big discoveries, and they didn't know if they should give the prize to Ehrlich or to Pasteur was dead at that time, to Ehrlich or to Metchnikoff. And so what they did, they shared the prize, and in 1908 they shared the prize between Ehrlich on the one side and Machitov on the other side, and they didn't say for this type of immunity or that type, but for their contribution to immunity. So that was fun. And then all these people, including Bering, died around 1915, 1916, and those pioneers were not there during the next decades. And then there were important contributions, namely from Jules Bordet on the complement. And complement again would be, but I'm not going to insist on that. That's dubious for those of you who are familiar with the story, because there's an input from both types of cells. And then there came a period of nearly 30 or 40 years where people were studying in detail the phagocytosis, how that happened in which types of cell, which tissue, and so on. And others were working much more on soluble factors in the plasma. And really, again, the watershed moment was after the Second World War, so that's nearly 50 years ago, when on the one side there was Gerald Edelman, he and Rodney Portner on the other side at Great Britain. Edelman was in the United States, when they succeeded, following the techniques of Linus Pauling, to characterize the antitoxins, which were named now antibodies, to characterize the protein structure of these molecules. And until that time, the controversy was still lingering on everywhere, but after that, after the discovery of those structures of the antibodies, protein structures of the antibodies, it was clear that that was clear to them, that was one of the central aspects of the immune defenses. Now, of course, well, you will be surprised to learn that, or maybe you know it, that at that time still, well, of course, in the immunity of Machitov, which was referred to by innate as innate immunity, the blood cells responsible were essentially called phagocytes, whether they were macrophages and so on. Dendritic cells had not yet been discovered. And still in 1970, the cells responsible for antibody production were not shown, were not identified. And that was thanks to Jack Miller in Australia and to Cooper in the United States, to that the lymphocytes were discovered first in birds, that was by Cooper, and then Jack Miller and so on. And then lymphocytes were identified, and it was rapidly realized, although that took more than 10 years, that there were two types. One type which was producing the antibodies, which Edelman had identified chemically, and the other was killing, referred to as cytotoxic lymphocytes. And those were then referred to as B cells, because the ones which Ed Cooper had found in the bursa, and bursa in birds, but there's no bursa in mammals. But in mammals, they're produced in the bone marrow, so they kept to the B. And the other ones were called the thymus dependent, so T cells. So B and T cells. So that was the moment when the really adaptive immunity was understood as being a very essential arm of the immune defenses. And interestingly, then of course the questions came, if the lymphocytes produce antibodies, well, then comes the specificity, and as we talked this morning about memory, and this then was a very essential question, and it was solved in the end by a certain number of people, like Niels Jerle, like Donegava, and so on, and Burnett, and there's a list of 6 or 7 people who did very important work. And finally, also, I would like to mention Mark Davis, and who cloned the receptor of the lymphocytes, and Mark Davis. And so, at that time, the whole picture of immune defenses had changed. And then everyone thought, actually, the real response is that of the lymphocytes, which is, as it has memory, can adapt to the first aggressor. So this is the adaptive immunity or acquired immunity. And the other story of innate immunity is not really that relevant. That's an ancient system, and so on. Okay, and now I will make a step forward. So I do not want to put myself into the list of all those pioneers, but I would say when I joined the laboratory in Strasbourg, I joined the laboratory where they're working on insects. And they were my supervisor, Professor Pierre Joly. He used to work on grasshoppers. Why grasshoppers? Because they are very aggressive destroyers of human foods. And France at that time had, as a quote-unquote, responsibilities in Northern Africa. So they were trying to, the government was trying to help identify models of reducing the number of those parasites, or not parasites, but those pests. And so he had worked until that time, he had worked on development and reproduction of grasshoppers. And when he was looking for a topic for me, he was saying, I have done all my work over 30 years now without ever using asepsis and or sterilizing tools with which I did the surgery. And I've never seen appearance of the infection. So insects must have powerful defense reactions. And would you take that? The mechanisms are unknown, he said, which was wrong. And then he said, would you like to do that story? And that was how I got involved myself in the story, and my colleagues, many of my colleagues after that. And why is he giving this story? Because it was very difficult to convince at that time. You imagine you have a great person, all this group of people who have shown lymphocytes in mammals, up to the receptors of antigens in mammals, production of the enormous amount of various antibodies, and even showing that one lymphocyte produced only one type of antibody. So everyone is excited about that. And you come and say, well, we are interested what the grasshoppers do. I said, come on, we don't want to do that. So that was part of the tragedy of my first years of getting funded. And then things evolved, of course, at a given moment, and that was when we had access in the 70s and then in the 80s and later on, of course, we had access to molecular biology. And then molecular biology allowed us to look at what were the receptors and how the receptors of innate immunity, I should say, invertebrates have no, they have only innate immunity, they have no lymphocytes. I should Am I too long? I'll make it short now for the rest. So just to explain to you that it was an enormous fight, which took 30 to 40 years for our group to convince people that they should fund us. And oh no, come on, that's not immunity, that's antimicrobial defenses. And then, again, as I like to say, there occurred several watersheds. And one was when it was rediscovered that in addition to phagocytosis, invertebrates, and namely insects, would also produce soluble factors. And there was a man whose name has been forgotten. He worked in Lyon, and it has not been forgotten, but I don't find it right now, but I'll find it before I finish this. Huh? No, Payot. Yes, Payot. That's a young brain, you see. Doesn't forget. Payot, yes. And he was working in Lyon, and he was really, it never penetrated, you see, the community. And then, when we had access, and then we found in the 80s, we found also, and Hans Boman in Stockholm found even a few, 2 years before us, there were molecules which are antimicrobial, which he called antimicrobial peptides. And then the field that was sort of a watershed in the history. And then people started saying, well, so, but those are present in insects. No, not only in insects, because now we're speaking about 1990. Bob Lehrer at UCLA in California and Tom Gans also there found similar molecules. And even funnily enough, they found that humans produced rosomycin, which was the hallmark molecule of what we had discovered in flies this time. So that was another watershed occurred. So we had the molecules, we had them in our hands, we had cloned them, we had the sequences, but of course we didn't know how they were induced. The relationship between the injury and the recognition of bacteria and then the induction. And then we were very lucky that independently, totally independent from us, the former PhD chief of Dominique Ferrandon in Germany made a series of tests to find out the sequences of genes which were controlling early embryonic development. And in that system, they found a certain number of molecules, which were initially only known as mutants, and then there was the peptide sequence was then shown, and that was the important thing. Was I remember nearly the day when that happened, when we learned that one of the genes encoding a protein, which went by the name of Toll for the phenotype of the gene, of the mutant gene, that had upstream sequence, the gene had upstream sequence, which had a site which had been reported several years earlier by David Baltimore, who got the Nobel Prize in '75 for his work on the retroviruses, and what he found it in B cells, and as a nuclear factor, they call it nuclear factor NF, in B cell and genes for kappa chain. I've met David many times since, and he always says to me, I would never have thought at that time, I co-authored it with a man named Sun, that this would become so important. And indeed, this site is mandatory for the induction of the cascade of genes controlling the very early cascade of dorsoventral patterning. Okay, we had by that time we had seen in the upstream sequences of the genes encoding some of our antimicrobial peptides. I say our because of the ones which we had found in the lab, and there was an NF-kappa B binding site in the upstream sequences, and then we said, well, that's a sort of great moment. Wow! So this must be. And then I presented this first data at a memorial lecture in Bern for Professor Lüscher, who had died a year earlier, and I presented this, and then there came questions, and then someone stood up and offered classical way of pretentious young person and said, I'm sorry, you're not a Drosophilist, you don't realize what you're speaking, you're comparing now early embryonic development, and you're working on adult flies. Come on, that's not the same system. Okay, I remember this not dearly, but I remember it's because my daughter and my wife were in the audience, and they were so embarrassed. And of course, I didn't have the answer ready, but then this was one of the last incidents. Then rapidly, many people jumped into the story, and then it came in a matter of 5 to 10 years, so the things became clear. So the receptor of innate immunity is controlled, one of the receptors, but a central receptor, I must say, is controlled by a cascade of genes which include NF-kappa B binding sites. And but still, in the early 90s, I had difficulties in getting the laboratory refunded by our CNRS, because the director of the CNRS funding agency, he said, well, stop doing that. We did also work on egg Dyson, and said, continue egg Dyson, you are known for egg Dyson studies, but this is not an immunity. This was 1990. This is not immunity, this is just antibacterial defenses in insects, and who cares about insects? Okay, and then from that, I mean, the rest everyone knows, I mean, how things evolved from there on. I was just trying to present how things started for us and for many other people at that time. And so, would that be enough, Madame?
Thank you very much.
It was too long, I'm sorry.
No, it's very good. I was about to ask you how did you discover the role of toll in the immune response, but you already answered most of it. But then the second part of the question is that how do you think this discovery revolutionized the understanding of innate immunity at that time?
Yes, but that's a very good question. I should have answered that also, because what really made the world drew attention to us was a moment when. So we found this story, and that was in '96, and then the year later, Medjitov and Genoway, with whom we had collaborated, to whom I had shown the fly overgrown by the mutant fly overgrown by fungi, they found in a cell culture system by homology screening, they found that there was in the genome a molecule sort of identical to, well, homologous to the NF-kappa B molecule, to which we had, I'm sorry, not to the NF-kappa B molecule, but to the receptor which we had shown in the fly to control expression of innate immune genes. So, and then the year after, Bruce Beutler, who gave his presentation, then he had been working for 5 years to find the induction of the gene, controlling induction of TNF-alpha. And again, he was going through, he was working, I should say, but maybe some of you know that, on the TNF-alpha induction by lipopolysaccharides, because that was the system which they used at the time. And finally, he found, after 5 years, as I said, he found a gene area which, when mutated, blocked inducibility. It was in a mouse which had been reported 20 years earlier to be resistant to infection by gram-negative bacteria and to injection of lipopolysaccharide. And that, even he phoned to me in Strasbourg, so we found it, and it's a toll-like receptor. It's a toll receptor. Actually turned out to be TLR4. So that had shown that there was a full pathway in the fly which directed production from an antimicrobial peptide. It was also present in, well, the receptor was also present in the fly, and then, so in humans, in the fly, and then came the important discovery by Ralph Steinman, who had worked on a cell type which was ignored by nearly everyone for 20 years, which they called dendritic cell, and then he showed that this activation of dendritic cells required the induction of innate immunity by the toll-like receptors. And so it was a whole story then, and that finally, and then the attention was, so and that changed, I mean, it changed everything. And now, of course, it should be said, not that I'm a particular fan of toll receptors, but you have heard in the various talks, you have heard that in humans, there are a dozen, let's say, 10 toll-like receptors, and we recognize a large variety of inducers. And so, but they're not the only. Until the year of 2000, in the meetings which we had, we all were convinced that that was a solution. And then people like Jean-Luc and others came and showed that there's C-gasting pathway, and then Dominique, that there was peptidoglycan recognition proteins, glucan binding proteins, and so on. And then it turned out that now we will, I don't know, we will maybe fight one day, which is the most important receptor, but we will not.
Thank you, Professor Hoffmann. It's very informative and very encouraging, inspiring story, particularly for the young scientists. Now I have the question still from the younger scientists to the senior scientists here. Why do you think that fruit flies, particularly in this case we're using Drosophila melanogaster, considered an ideal model for studying immunity? And what specific features of the immune system make them so valuable in your eyes?
Okay, so I will start answering the first part of the question, and I guess I will let Jean-Luc deal with the second part. So first of all, I'm not sure there is an ideal model. There are always limitations to the models, and one of the limitations of the fly to understand innate immunity is that they are cold-blooded, and there is no heat that is actually a very strong protection against many microbes. That's one of the limitations we have, even though there is behavioral fever, so the flies, when they are sick, they tend to go to warm places. And That's not the major reason. So, first of all, the history of Drosophila is very long because it was started basically from the time of Morgan and some predecessors, so on the turn of the 20th century, and with the introduction of genetics. And what made it also a very good model is that the flies are small, they are cheap to raise, easy to raise, and very resistant to infections, so there are not so many problems there. And also, what we have is this inheritance of more than a century worth of genetic studies, leaving really us with very potent tools, and that have been optimized over the years with modern tools of molecular biology. But already, the basic tools that were done by classical geneticists were already giving a lot of power and enabling my PhD advisor, Professor Nusslein-Wollard, together with Eric Wischhaus, to ask some very essential questions that nobody would have dared asking. And their question was, what are the genes required for early embryonic development? You know, it's not a small question that you can ask. And the answer was actually to use unbiased large-scale genetic screens. So using chemical mutagenesis, so like this, you're really unbiased because it's basically random mutations that you are generating. And then you're basically going to generate so many mutant lines that at some point you will have it each gene of the genome on average. I mean, of course, there are always some exceptions, some very small genes that are difficult to target, but that's the notion of saturation. And basically, when they were doing this screen, that they used actually on what are called zygotic genes, so the ones that are required to make the embryo after fertilization, and once the embryo starts expressing its own genes, that was just for them a proof of principle. That was not the question they were really interested in, but nevertheless, they found this way 400 genes that were actually perturbing the pattern of the early embryonic larvae afterwards. And they could really see with this very potent way to screen, you could get this. And during these studies, they actually found the first mutant of Toll, which was dominant, and then it was really this question. So I think, to me, when you want putting the questions, what is the strength of the Drosophila model, I would say genetics, genetics, and genetics always, because you can have these large-scale approaches and really asking the question in a very broad manner. So at the beginning, when we started working on innate immunity, at the beginning, Jules has reminded us that they used actually mutants that had been generated by the laboratory of Nusslein-Wollard and others, and that really worked it into a pathway. So part of it was conserved for the innate immunity, but that was really one of the things. And then what we did was just to ask, okay, can we directly address the question of innate immunity using unbiased screens? And we had some biases at the beginning because we were using reporter genes that were just using effectors of the two immune NF-kappa B pathways. That helped to find out quite a few genes in those pathways. And then we had second generation screens where we were using this time a transposon insertion line. So we knew already from the beginning which genes we were actually targeting. So these were not unbiased, but they were semi-targeted from the analysis of the genome, which was a major. We have to remember that the genome of Drosophila was sequenced in about 2000, and that was the second metazoan genome, second only to the small nematode worm, Cenorhabditis elegans, that is also a strong workhorse of genetics. So it was very, very important. And so from this, we could really find the genes that are involved in innate immunity. And I will hand the microphone to.
Thank you, Dominique. Yeah, from my point of view, when I joined the lab in Strasbourg, my experience in immunology was in mammalian models. I worked on regulation of immune responses by cytokines. I did my postdoc at the DNEX Research Institute, where Th1, Th2 cells were discovered and their regulation by cytokines. And the situation was that when you started to do genetics in mice on these molecules, is that there's a lot of redundancy, and so the phenotypes were not that clear. And so to me, the first reason was the simplicity, that because you expect things to be more simple in flies than in mammals. And of course, this is a conception that people often have, and still nowadays, that when you mention a pathway, the reaction is, oh, this pathway, the flies already have it, as if we derived from the flies. But in fact, flies, insects evolved separately from vertebrates. And so the pathways that are in insects have been evolving for as long as the pathways that are on the mammalian side. And so that's the irony of it, is that now our work using the genetics, trying to stay as unbiased as possible, as Dominique insisted on, led us to find the important role of the C-gas-sting pathway in antiviral immunity in Drosophila, and our work shows that this pathway seems to be more complicated in flies than in mammals. So we have to be careful about the preconceived ideas, and this thing that it will be simpler in flies is not necessarily true. But then what is definitely simpler is, in order to carry experiments, it's much more simple to get the authorizations and to get the funding, because it costs less than to keep mice stocks. This is a definite advantage of the fly model. And this said, if you, as Dominique mentioned, another very popular invertebrate model, which is the nematode, which would be even cheaper to use. So what we're fortunate with the flies is still it has a complexity. Maybe our friends who work on nematodes would not be happy if we said about that, because actually nematodes have more genes than insects, so there is a complexity in nematodes. But when we consider immunity, there's a lot of key pathways that are present in both insects and mammals, and that appear to be lacking from nematodes. For example, there's no NF-kappa B in nematodes, and so this is something that is interesting with Drosophila. A last thing for me, who works on, and for us, with Lauratea, who work on viruses, another interest is, of course, that some insects transmit deadly viruses to humans, and so the fact that we can quite rapidly test the relevance of a finding in flies into mosquitoes is important. But this said again, when you look at the genome, it seems that the mosquitoes don't have C-gas-sting, so even though these are both dipteran insects, there could be some differences.
And here is a question for Professor Brandon. Following the Professor Hoffmann's pioneer work about fly innate immunity, many achievements have been made to understand how the fly responds to the pathogen infection. And here is including the discovery of Tor, MD, and Jack State, dicer, and the C-gas-sting pathways. And here the question: how do the fruit fly innate immune response adapt to chronic bacterial or fungal infections, and what insights does this provide for the understanding immunity tolerance or resistance?
Okay. Good, so maybe we should start back from where Jules started. So the effectors of the classical antimicrobial response against bacteria and against fungi. So this was the identification of some antifungal and antibacterial peptides, and the fact that they were regulated through these NF-kappa B binding sites in response to infection. And that was really a starting point, and I will come back to this later. I will come back to these effectors to give a complementary approach also to what the function of these induced molecules are. And so, to make a long story short, so we have two immune pathways. The immune deficiency NF-kappa B pathway that is mostly triggered by exposure to gram-negative bacteria. And specifically, what is really important is peptidoglycan as a main component of the cell wall. And the second pathway is the DOL pathway that we have been discussing already. And this one is required for us defense against, very surprisingly actually, gram-positive bacteria, so that means prokaryotes, and at the same time against fungal pathogens, which are actually eukaryotes, and with a very different biology. So it's really some things that we don't fully understand, although I think we are starting to get some ideas. So for the immune deficiency pathway, through genetic screening, I mean, works that had been done, for instance, by François Lelier, was really suggesting that peptidoglycan was a key molecule, and at the time, everybody was considering lipopolysaccharides because of the mammalian field, and there it became very important actually to use very clean reagents to be sure that, I mean, if you were buying commercial LPS at the time, it was full of peptidoglycan, and if you are using peptidoglycan, it was contaminated by LPS. So it was it was sometimes a little bit difficult to deal with it. But basically, we had this candidate gene approach that allowed to actually discover that one peptidoglycan recognition protein that is actually membrane-bound, PGR-PLC, was a major member of the IND pathway, and that it was basically mediating. And then after it went very quickly, because then we had structural data that were really showing actually all the peptidoglycan recognition proteins were actually binding to peptidoglycan using different isoforms, different combination of isoforms, and I don't want to go too much into the details. But one of the things that is also important to consider is that when we manage to have bacteria inside the fly, provided you're not putting massive amounts, you do not necessarily immediately trigger an immune response. And actually, the system is very much tuned to actually not detect really the bacteria themselves, but the fact that they are proliferating. Because when they are proliferating, they are dividing and they are remodeling their cell walls. And when they are remodeling their cell walls, they are releasing small peptidoglycan fragments that are uniquely sensed by the PGR-PLC and PGR-PLE receptors, so this family of receptors. So which means it's a notion of a PAMS per vita that was developed by Russell Vance that you're also detecting the life events that are there. As regards the detection of gram-positive bacteria, we found that the second category of proteins are important, so they are called, and this is a misnomer, gram-negative binding proteins, so we just call them GNBP, because they contain actually beta-glucan recognition domains. So they are really more recognizing metabolites that are made by fungi than anything else. But also sometimes you can find some circular beta-1 free glucans in the periplasm of bacteria. And there was another very important receptor that had been identified by our colleague Julien Royer, and that was again this time of the small peptidoglycan recognition protein. And the difference in this case, the toll pathway sensors in this case are circulating receptors that are being secreted and that can detect the respective type. So actually in terms of difference between gram-positive bacteria and gram-negative bacteria, it's the quality of the peptidoglycan that makes the difference. So gram-negative bacteria have mostly a type of peptidoglycan called diamino-pimelic type, so DAP type peptidoglycan, and many of the bacterial pathogens, gram-positive bacterial pathogens that we have are actually with a lysine type, and these are better sensed by PGR-PSA in conjunction with GNBP1, and we could make actually transplant experiments and really to demonstrate that actually PGR-PSA and GNBP1 function together in sensing bacterial infections. And then, as regards fungal infections, we identified another member of the GNBP family and showed that it was actually able to bind to beta-1 free glucans that are a major component of the fungal cell wall. But this was nevertheless not a whole story because in some cases, we had some kind of paradoxical results where we did not see really a phenotype when we are killing GNBP3, and this is really the contribution from Petros Ligoxigakis and Jean-Marc Reichert in the laboratory. They had identified this protease called Persephone, and this one is really a sensor actually not of the structures of the cell wall, but actually of the virulence factors that are secreted by pathogens. And in this case, proteases. So they are really acting as a bait for proteases, so the story is a bit more complicated. But basically, you had really this way to sense, and actually, all of this is taking place in the hemolymph. And what you have is that you are actually going to activate proteolytic cascades. So cascade means amplification, and all of them will finally culminate in converting a procytokine named Spätzle, that's the German for noodles, just because of the embryonic phenotype formed during these developmental studies. And once you cleave a pro-Spätzle into active Spätzle, it becomes an active toll pathway ligand and will trigger the canonical toll signaling pathway. So now in terms of antimicrobial effectors, it's surprising that even though we had identified several antifungal peptides, the work from Bruno Lemetzlaff has shown that they are, and also from Steve Wasserman, have shown that they are not the main effectors of the toll pathway. But the main effectors are actually a family of peptides called bomanins. And what we have found is that, this is not published yet, that some members are pretty much active against gram-positive bacteria. At the same time, they have also other actions. And one of the actions we found, and that is really protecting, especially in the case of Aspergillus fumigatus infections, is that some of these peptides, they are not directly targeting the pathogen, but what they are doing is actually to protect the host from the action of mycotoxins or secreted virulence factors. And they can, in some cases, prevent the action of these toxins, or in others, actually repair and enhance the repair, so allowing the fly to endure. So this is what we called, what I call resilience, and then also in the field as disease tolerance. So you really have these immune effectors that are so important. They are not only targeting the pathogen, but also enabling the fly, the host, to deal with these toxins.
Thank you for Professor Brandon's insight about the antibacterial and antifungal immunity. Now we move to the antiviral immunity in fly. There are many unknowns about the antiviral immunity, such as we have discovered the nitrogestate and the C-gasting pathways, but there are still many unknown. Professor Imre, can you share with us your insight about can fruit fly models help to identify novel antiviral agents or therapeutic strategies that might be applicable to human diseases? Thank you.
Yes. So yeah, in terms of if we look back now, we've been working on antiviral immunity now for many years in the flies. And so if we look back, there are several mechanisms which are involved in the control of viral infection, but there are two that stand out as particularly important. So one of them, which I believe is the front line, the first line of defense, is RNA interference. So obviously this is very important. And then there's induced responses which involve the C-gasting pathway. And what's interesting is that at the same time, in mammals, the antiviral immunity is dominated by the inducible response regulated by interference. But there are also reports that RNAi is also involved, but it's more discrete and it's probably only in some cells where the interferon response is not operative, which suggests some kind of incompatibility between the two mechanisms. And so I think one question we have now to address in the flies is to understand the relationship between RNAi and the C-gas-like pathway, because they seem to be activated by the same molecular pattern, double-stranded RNA. And so the question is there a hierarchy between the two pathways? Is one of them involved in some tissues, the induced response in other tissues? All of this we don't know. And it would be interesting to see, to have answers to that. And this might be also relevant for mammals to understand why RNAi is sort of not more important in mammals. So now, when it comes to the help to identify novel antiviral agents or the relevance for work in mammals, I can see two directions. So the first direction is, as I was saying before, the C-gasting pathway seems to be more complicated in insects as mammals. And so this might be a consequence of the fact that insects evolved on different paths from the other animals. And so they were confronted with needs that created this diversification within the C-gasting pathway. But another thing is that it might allude to the fact that the C-gasting pathway is maybe more complex than we think in mammals. And maybe there's another dimension that has not been seen yet and that exists there. In particular, our collaborator Philippe Kranzusch at Harvard has crystallized another molecule of the C-gas of the C-gas family, and clearly it has all the feature of a functional C-gas. So it's just that for the moment, nobody has found a ligand capable to activate it, and so nobody could monitor the production of a product. But maybe there are other C-gas receptors in mammals. So that would be one aspect. So learning things of the C-gasting pathway that we don't know yet in mammals. And the other dimension is, of course, characterizing the antiviral genes that are regulated by the C-gasting pathway. And this is one of the things that we believe is very exciting, because what we see is that in Drosophila we have some 200 genes which are induced by the C-gasting pathways, and among them there are antiviral genes. And these genes, I think the key point to understand is that we live in a common virosphere. So the viruses that infect all animals are present in the terrestrial environment. And of course, there are insect-specific viruses, but when you look at the viruses infecting insects, they belong to big families which contain members also infected mammals. So there are pox viruses infecting insects, there are picorna-like viruses infecting insects, there are flaviviruses infecting insects. So all these families have relevant members which are serious human pathogens. And so I believe that we can learn things from the factors that restrict these family of viruses in insects. We can learn things about maybe new strategies to develop antivirals for mammals. And so we have some 200 genes, including antiviral genes in Drosophila melanogaster. But when we started to probe other species of Drosophila, we find that each of them has 1 to 200 induced genes. And there's a core genes which are conserved in all these flies, but each species has a set of 10, 20 genes that are not in all species, that are taxon specific. So if you multiply that by the number of insect species, this gives you a lot of what we call innovations. And I believe that there is room to find very original antiviral genes.
Okay, now it's my turn. So thank you for all the three senior professors. Now we know much more about the contribution of Drosophila models in the controlling infectious disease. So recent years, we noticed that Professor Hoffmann, together with Professor Chen, extend your work from infectious disease to non-infectious. So my question is, how do Drosophila respond to cancer cells, like oncogenic cells? And how does the work in flies contribute to identify novel immune target for cancer immunotherapy in humans?
I'll pass the word to Professor Chen. Well, we work together, so it's, I mean, I have already spoken too much. That's certain.
Okay, thank you for the opportunity to share some of my opinion. Well, actually, when we started this work with Jules, who initiated in the very beginning the topic, when we started the work, we were often asked the first question, does flies will have cancer? And then this is essentially the question. The answer is yes, actually, they naturally occur cancer. And it was discovered earliest about 100 years ago by Mary Stark, who was a student in Morgan's lab. And then actually they naturally occur cancer. And then the question is, of course, now in the field, we already have some clues about how flies fight against bacteria and how the fly recognize also the viruses. But then how the flies respond to cancer if they occur. Will that be a reaction or not is still unclear. And actually, before our study starts, there are quite some groups already working on it. And they essentially, most of them are using the Lavi model, and they have gained some progress, particularly regarding some hemocytes. And a few of it also discovers that the JAK-STAT pathway is involving also the antimicrobial peptide. But I'm not going to put in comments for all that. But I would like to say that when we were starting, that there is still quite some unknown regarding how the flies recognize the oncogenic cells. This is very important. Just like how the flies recognize the bacteria and fungi and recognize the viruses. This question is largely unknown. So then, to answer these, we think of it, and then eventually we compare the advantages and disadvantages and decided to use the ADA model and establish a new, Drosophila tumor model based on injection of cells into these oncogenic cells into the flies. And then now we have some few discoveries. First of all, we find out that this type of reaction is totally different from what the fly is fighting against the bacteria and viruses. The whole transcriptome is totally different, actually, if you look at the whole profile. And then second interesting thing is that we find that there's some molecules that are very important at the early embryonic development. And they supposed to be silenced or relatively calm in the adult flies. But surprisingly, they reactivated or being challenged and reused by flies to fight against the tumor cells. So this is something very interesting in our study. So we are going to look deeper into that. And also, I think Drosophila as a model, it can at least give us some clues of in the future if we can we already, in our ongoing study, identify few receptors, actually, specifically responds to the oncogenic cells. And then if we manage to find a ligand, which we're doing now, then that will be a sort of like a conceptual proof of that there is a specific recognition for oncogenic cells in flies and bees. Yeah. We hope to make improvement in that in the future.
Professor Hoffmann, do you have anything to add?
No, it was excellent. Thank you. That was very good.
Okay, so next question is for Professor Renjie Zhao and Changxian Wei. So we know that Professor Zhao and Professor Wei continue to explore the potency of Jezebel models in other diseases like aging or central nervous system disease. So Professor Wei and Professor Zhao, could you please tell us about the crosstalk between the innate immunity and other biological systems like central nervous system or aging?
So probably I say a few words about the nervous system and immunity regulation, and I leave the aging issue to Changxian. Okay. Well, what we are interested, the organisms is kind of complicated, and the biological processes cannot be kind of normally purely independent from the others. One system from working totally independent from the others. This is my perception in my head somehow. Of course, we have been talking about innate immunity discovery and how it's working, especially in Drosophila. But I believe, and actually in the literature, there are also already papers stating that there are clearly interactions between the immune system and the nervous system. Actually, in the beginning, we are interested, wanted to investigate in both directions. Regarding the interaction between the nervous system and the immune system, of course, there are two directions. One is how the immune system affects the nervous system. Another thing is how the nervous system regulates or affects the immune system. Me, myself, and my group are more interested in the direction of how nervous system affects the immune system. And answering this question, we kind of wanted to address in two ways. One is how, or are there any, actually now we know there are specific neurons or neuron circuits that are responding to immune or to pathogen infections. This is a very interesting phenomenon, but it's also a big question. This includes how the signals gets to the, or how the signals activate specific neurons, and the activated neurons, how it passes the signals to the immune system to make the body to kind of cope with, together with the immune system, to cope with the invading pathogens. Well, we have already some findings. We did find specific neurons or neural circuits specifically responding to infections. And another part, we were wondering, is there any neuron-specific or neuron peptides, let's say, or molecules or peptides secreted from the neural system that works on immune response. We did also find at least one molecule that are responding and are involved in immune response. Regarding the other direction, how immune cells, let's say, how to protect or to interact with the nervous system, I think there are quite a lot of publications, especially those from the mammalian studies. That's kind of the function of the immune system, to protect the nervous system. Of course, the nervous system needs to be well protected. That's why there are so many people are interested in that direction. But this is not really the interest in my lab. Okay. The Changxian, would you say something on the aging and immunity interaction?
Okay. Actually, Professor Zhao has also a lot. I will add some my thoughts about this question. Actually, the aging is also a very complex process, or someone call it a disease. Aging, maybe someone called it a disease. It involves the functional decline of many tissues or organs in the host. And among them, actually the chronic inflammation is one of the hallmarks of aging. In the host, especially in the fly, the chronic inflammation refers to the activation of innate immunity pathways, such as the TOP pathway, MD pathway, and the CKST pathway. They produce many inflammatory molecules, such as the AMPs, that kind of thing. And there are also many reports. They have found the activation of innate immunity pathways can lead to aging. And during the aging, the cell death or the mitochondrial dysfunctions can also lead to the activation of innate immunity pathways. The innate immunity and the aging, they are intact with each other, and they promote each other's activation or the pathogenesis. In this area, actually for now, we know some, but there's still many things we don't know, such as the detailed molecular mechanism. This field still need further investigation. Actually, here this morning, after the meeting, I discussed with Dong Zhao, who was a postdoc of Bruno, and now he's a professor in Hunan Normal University. I proposed a term called immunophysiology. I suppose this term can describe the interaction between the immune system and with the other non-immune system, such as the brain, physiology. They including many things, actually.
Thank you. So last question is regarding the Drosophila genetic tools. So we know that one of the major reasons that Drosophila can make significant contribution to the biological research is the genetics. So, and Professor Jiao is one of the pioneer in gene editing using Drosophila. So Professor Renjie Jiao, could you please summarize the resources of Drosophila genetic tools that have been made, and how does the tools contribute to the research on immunity?
Well, this is a difficult question because I don't have a specific answer, kind of. Actually, from the time of Morgan, Drosophila has been known now for more than 100 years for the advantage of genetics for studies in cell biology, genetics development, neurobiology, and immunology. And actually, Dominic also just mentioned that Christian Nusslein-Vorhard and Eric Wieshouse, they made large-scale screening for early developmental genes and things like that. I don't know, are there anything specific for Drosophila, any genetic resources specific? Well, there are resources for genetic resources for Drosophila studies, but specifically for innate immunity, shall I say? Because innate immunity, you're studying a physiological process and gene functions or protein functions. This is a kind of normal strategy. We use genetic tools to study in terms of loss of function, gain of function, and in combination with biochemical studies. So I don't know how to say. Is there any specific genetic tools used only in innate immunity but not other studies? Probably not. Yeah, that's what I wanted to say. The genetic tools can be used in different disciplines. I could say just now we have some genetic resources. As you just said, gene editing. Me, myself, or my lab indeed have been following or have been interested in developing or applying these genetic manipulation techniques. Actually, starting something like more than 20 years ago, when the first knockout techniques was, which was developed by Dr. Yi Tangrong, which was published in 2001 or so. Because before that, to make loss of function Drosophila strains, it's basically it was basically dependent on other strategies like PLF and jump out, like non-specific mutagenesis. yeah, or some other things. And starting after Dr. Yi Kangrong developed the specific knockout technique. This is actually kind of unusual because this technique was this specific knockout technique was after the mammalian system. Yeah. So the reason you know that because in Drosophila germ cells, especially the male germ cells, it's difficult or basically nothing. There are no homologous recombination there. That's why this homologous recombination based gene targeting was not possible. But anyway, Dr. Yi Kangrong, he kind of had a kind of idea that to linearize the exogenous targeting plasmid, then which increased the homologous recombination very much. That's why he made it possible for Drosophila people. And after that, there were coming gene targeting techniques based on zinc finger proteins and afterwards TALEN proteins. Actually, my lab has been also involved in these techniques. Yeah, we published, I believe, the first TALEN-based gene targeting technique in the world, and that's 1 year before the CRISPR-Cas9 technique. Then, if there would be no CRISPR-Cas9 technique, the TALEN technique would be, well, possibly, yeah. Anyway, that's how it goes. Anyway, now it's very easy, basically, for every Drosophilist who wants to make it on his own or her own. It's very possible. But in my lab or in the institute of SFHI, actually we are using CRISPR-Cas9. We have made a collection of more than 5,000 genes of mutants. So which is, of course, a very good source for both innate immunity study and other studies of Drosophila. Okay.
Thank you, Professor Zhao. Now I pass back to you. Okay, I'll be chairing the last section. So there are still a few questions for I would like to ask the senior scientists. What do you think remain unanswered? Important questions in your eyes in innate immunity. What do you think?
I'm not senior. Well, we learned during discussions here that it is becoming extremely complex to understand how the various pathways concur or are interrupting the other. So there are many unknowns still. Jean-Luc just mentioned that they have about 200 genes which are expressed under the control of this or that transcriptional activator. And you have in your work, you have hundreds, if not 1,000 and some, and we have no idea what these molecules do. And we are centered on looking if they have a phenotype. Okay, that's probably the easiest or the most spectacular thing if we find a phenotype in development or survival and so on. But the interaction between those various, you just imagine that in the cells you have this crowd this crowd of molecules, and they will interact. Even, as we know, something which would seem simple now, like mid-88, that controls other toll, depending on the activator of toll and the complex this form. Excuse me, can have totally different effects in the physiology, and all this is still unknown. And it's for the fly, it's a pleasure to study that. But in human beings, I was really surprised of. I should have known it. That if you have various diseases, various cancer diseases, and so on, and then we see it now in the very simple way that this is not regulated, this is not regulated, and yes, and so that's. What we call simply for the time being an off-target of this medicine can be extremely important. We don't, for the time being, we don't know much. And I wouldn't know how simply out of increasing the funding, increasing the number of people who work and so on, but that will probably never really happen. So how we are going once over maybe 1 century to understand what is really going on beyond what we have seen. I mean, Nisha and Paula did beautiful work, but they were looking at spectacular phenotypes. You see. But if we go down in simple metabolic phenotypes, so I would say that's one of the things which there are other frontiers which I would see, and I would even say it scares me.
Okay, Jean-Luc and Dominic, you have something to.
Yeah, I fully concur. There's still a lot of things to discover. I mentioned earlier I did my postdoc at DNX. I want to mention the president of DNX when I was there was a very nice man called Al Waits, and DNX was operated by the Sharing Flow Corporation. They were paying all this, and Sharing Flow made its fortune with antibiotics. And he was a scientist. He had discovered a couple of antibiotics, so it contributed to the fortune. His reward was to end his career in California in this institute. And he retired quite young to go fishing in British Columbia. He was a big fan of fishing. And I remember we discussed at his going away party, and because he was always very curious about science and very fun to discuss with, even for postdocs. And his point was, so the question was, okay, won't you miss science? And he was, the major discoveries, because of course he had witnessed, all these things that Jules mentioned, the discovery of antibodies, gene rearrangement, all these things, and it basically was, okay, yeah, we will find a few more cytokines, and but now we nearly. Okay. And then came RNA interference, and then came innate immunity, toll-like receptors, pattern recognition receptors, and I think there's still a lot of things and that's the beauty of basic science, a lot of things that we don't even suspect that are there. And if I have to make guesses, I think some of the big surprises will come from integrating things, so the complexity, all the gene interaction things. I think this is a major challenge. And now with artificial intelligence, I think that with all these techniques emerging, that comparing things by looking at protein structure rather than sequence homology, this opens lots of horizons. And this challenge of integrating things, how all these pathways that each of us studies separately, how all these things integrate into a living organism, I think this is one of the added value that will emerge in the coming years for the flies. Because at one point we had some discussions even among us about funding, because a lot of things that were possible, that were the assets of the Josephina model, like genetics, in fact, they became available in mammalian systems, because you can do genome-wide screens now with CRISPR-Cas. But the thing is, you do it in cell lines. So you lack this integrative dimension of the live organism, and I think the flies have that. And I think if I want to conclude with the SFHI and the future of SFHI, indeed, like key questions is how the connections between anti-cancer, like non-infectious, like anti-cancer, aging, and infectious things. There is obvious interactions, and I think at SFHI we have the conditions to, by just letting the people in charge of the different group work freely, giving them enough money to work, what they will find will be integrated. So I don't think there's pressing need. We don't need to bother ourselves about, okay, what is pressing for translation into human medicine. If there is anything, that the translation will come.
Thank you, Professor Imla.
So there are two aspects I would like to develop a little bit. I think one of the major challenges is that we have to understand infections, and each infection has its unique features, depending on the pathogen and the host. And now we have come at a level. Yes, good. So we have come at a level of understanding of the basic mechanism of innate immunity in the flies that it becomes very meaningful to study host-pathogen relationships in detail. So for instance, from our own studies with both fungal and bacterial pathogens, we see that the route of infection is very important, and that depending on this route, the pathogen will express different sets of virulence factors, and also how it's reacting to the host immune response. So, for instance, Pseudomonas aeruginosa is a very potent virulence factor, very potent pathogen, and it can kill the flies in 48 hours. But if you feed it to the flies for a brief period, it's still able to escape the gut compartment and go into the tissues, but it will not be virulent at all. And we have found that actually what actually drives the downregulation of the virulence is an insect-specific immune response called melanization, but we need to understand exactly how at the molecular level the bacteria are coaxed into avirulence because of this. So this would be a new strategy to fight with infectious disease. The second point is that Drosophila is very good for developing novel biological concepts. I think even if the effectors are not evolutionarily conserved, it can point to the good directions to look at. And I will come to one of my favorite subjects, which is fungal infections. And I think we have a real subject here because there are really emerging diseases that are going to be very threatening, not only to us ourselves as humans, but also to the biotope. So for instance, we have had catastrophic diseases eradicating frogs, bats that are very prevalent, and we are totally left without arms. And then I just want to point out also the effect of global warming that I discussed this morning a little bit. Temperatures are increasing, and so our best defense against fungal infections is temperature. And the difference is decreasing, and the fungi are progressively adapting to higher temperatures. So that means that there are going to be major pathogens sooner or later. And actually, we already have this problem with Candida auris, for instance, that is becoming a major pathogen. And the second aspect is, for instance, toxins. So fungi are really secreting a lot of toxins. Some of them are volatile. And what do we have? We have global warming nowadays. So we get all of these catastrophic floods. We had one very recent event in Spain earlier last month. We had them in France, in Germany, in Australia, in the United States, in Guangdong. And for instance, when I came here a couple of weeks ago, what was happening in Hainan, they were weathering their 24th typhoon of the year. Okay, and what you have to know is that, of course, this brings to high humidity, and these are perfect conditions for the growth of fungi. And once you have your house flooded, the fungi will colonize the walls, and you will never get rid of them. So that means that all of the time you're going to get exposed. And I know even here in Guangzhou, where the weather is very wet, one of my postdocs had to change our pillow. It was a PhD student, so because fungi were growing in it. And the closest of the son of one of my assistant professors, there was also fungus growing. And that means that we are going to get more and more exposed to these mycotoxins. It's very important to understand how the human body is able to deal with these mycotoxins.
Thank you. Thank you. So one last question I would like to ask, particularly Hua Cai. In what kind of technology you are specifically interested in, and you think it would be very useful probably in the future for our study?
Well, it's a difficult question. Why specific for me?
No,
yeah,
just
so yeah, in Drosophila, from my point of view, there's a huge space to improve the technologies. So, for example, the spatial transcriptomic and more accurate than mass spectrometry, and just the reason is this is the data I think from Jules, like 80% of the medicine that the human invented actually isolated from the plants. But actually, if you look at the number of species of insects to plants, insects they have more species than plants. By insect, there are around 1 million species. For plant, I think is only 1/3 compared to insects. So, and insect has a very long history of medicine in China, like 3,000 years ago. There's around more than 10 insects can be used to treat some diseases. And now, based on the Chinese government, there's around more than 400 insects can be used for treating different kinds of diseases. So there is a lot of active compounds in insects, including Drosophila. And the Drosophila species, there's 1,500 different Drosophila species. So, and to isolate those active compounds, we need a very accurate mass spectrometry to identify active compound. And those compounds probably can be used for medicine in future, just like the compound isolated from the plants, right? So, and we work on innate immunity against no matter infectious disease or non-infectious disease, but all the compounds we identified which involved in this process can have a potential to be used for human diseases. So I think mass spectrometry could be one of the technology that need to be improved. And another thing is like spatial transcriptomic, and not only transcriptomic, also spatial metabolics, and those can let us to better understand about the tissue communication as a whole, because the Drosophila, one of the major is in vivo assay compared to cells, because they have several tissues, although it's tiny creatures, but they have entire different systems, central nervous system, digestive system, and reproductive system. So to understand the physiological as a whole, so we need like spatial technologies to get better understanding about how tissue communicate each other. I think this is also very important for the future. But next year, other not all space, I mean, in terms of technology, Drosophila could be improved. So, but we can think about that. Yeah.
I think in your study you're also looking into, the molecular structure prediction, protein structure adopting.
Yeah, especially in the big data.
It could be important for the immuno across the species.
Sure. Yeah.
Okay. Thank you. So that's it.
Yeah. So finish.
Yeah.
Thank you for
all of you.
All you senior professors.
Participation and your time and yes.